I want you to picture something really specific for a second.
Okay.
You are standing backstage before giving a massive presentation. Or, maybe you're sitting in your car right outside an office building before a really huge job interview.
Right. The high-stakes situations.
Exactly. Your palms are sweating, sure. But what is the defining physical sensation right in that exact moment?
It's the stomach.
Yes. It is that fluttering, restless, almost nauseating feeling right behind your belly button. You know the exact one.
Yeah.
Classic butterflies in your stomach.
Oh, absolutely.
Or think about the exact opposite scenario. You're walking back to your car after a nice dinner, you reach into your pocket, and you suddenly realize your wallet isn't there.
Oh, that is the worst feeling.
It's awful, right? It's
Yeah.
an instant heavy pit of dread. And it doesn't drop into your chest, it drops right into your gut.
Yeah, right.
We use these phrases literally all the time. We tell people to, trust their gut, or we say, I just had a gut feeling about that guy.
It is so deeply ingrained in our language, isn't it? We treat the stomach as this kind of poetic metaphor for human intuition and primal emotion.
Right? A metaphor. But what if we've had it completely backwards this whole time?
How do you mean?
What if that physical sensation of dread or that flutter isn't a metaphor at all? What if your gut is literally, biologically, a second brain that is actively processing the world around you?
That is a massive question.
It is. And that is our mission on the Deep Dive today. We are going to completely dismantle the way you think about your digestive system.
I love it.
We're going to look at how your gut works, not just as like a mechanical plumbing system trying to process your lunch, but as a highly sophisticated electrical grid and a massive chemical factory that directly controls your mood, your health, and literally your daily life.
It really requires a massive shift in perspective, to be honest.
Yeah.
Yeah, because we are so conditioned to think of the brain inside our skulls as the undisputed CEO of the body.
Right, the brain is in charge.
Exactly. The brain gives the orders, and the body blindly executes them. But when you actually look at the anatomy, really look at it, the gut operates with an unbelievable degree of autonomy.
Unbelievable how? Like, how independent is it?
Well, for one, it is the largest endocrine organ in the human body.
Wait, really? The largest?
By far. It produces the vast majority of our hormones, and it possesses a localized nervous system so profoundly complex that it can actually function entirely independently of the spinal cord and the brain.
Entirely independently?
Yes. If you were to somehow, hypothetically, sever every single nerve connection between your brain and your gut would just keep digesting, adjusting, and firing on its own.
See, that just sounds like science fiction to me.
It does, but it's pure physiology.
Well, to get to the bottom of how this is even possible, we have pulled together a really diverse stack of sources for today's discussion.
We really run the gamut today.
We do.
Yeah.
We're looking at foundational physiological textbook chapters. We have these incredibly detailed clinical profiles from stat pearls, covering everything from, gastrointestinal motility to specific cellular hormones.
The stat pearl stuff is incredibly granular.
Oh, it's dense. And we're also pulling from research by Kaiser Permanente on how the gut directly affects mental health. And we even have some historical medical archives to map out the dramatic, honestly, slightly messy way we discovered all of this in the first place.
And to really understand how the gut pulls off this autonomous control, we have to start way, way back.
Like historically back.
No, biologically back. Before you can understand how your stomach controls your mood today, you have to understand how it was built before you were even born.
Oh, okay. We're talking embryology.
Exactly.
Because I was reading through the textbook sources, and this system starts forming incredibly early. We are talking about the 3rd week of gestation.
Just 3 weeks.
Yeah. You were just an embryo, barely the size of a pinhead, and your body is already laying down the foundation for this second brain.
That 3rd week is a critical window. The embryo goes through a process called gastrulation.
Gastrulation.
Right. It's where it organizes itself from a simple ball of cells into 3 distinct germ layers. Think of them as the 3 primary manufacturing materials for the entire human body.
Okay.
You have the endoderm, the mesoderm, and the ectoderm.
So how do those 3 layers turn into the digestive tract? Do they just, fold into a tube?
Basically, yes. The endoderm, which is the innermost layer, folds to form the inner epithelial lining of the gastrointestinal tract.
The inner tube.
Exactly. It also buds off to create the associated glands, the liver, the gallbladder, and the pancreas.
Got it.
But a delicate inner lining isn't enough to physically push food through the body. So the mesoderm, the middle layer, wraps around the endoderm tube.
Like putting a sleeve over it.
Exactly. The mesoderm gives rise to the heavy machinery, the connective tissue, and the thick bands of smooth muscle that actually form the walls of the gut.
Okay, so that leaves the ectoderm, the outer layer. Usually that makes the skin and the brain, right?
Correct. But here is where the concept of the second brain actually physically begins.
Okay.
A very specific population of cells from the ectoderm, called the neural crest cells, actually migrate away from the developing spinal cord.
They move.
They literally crawl through the embryonic tissue, down into the developing gut, and embed themselves directly into the mesoderm.
Wow.
Yeah, and those migrating neural crest cells become the enteric nervous system.
They literally migrate down to set up a localized command center.
That's exactly what it is.
And by the time you're an adult, that little tube is grown into roughly 30 feet of incredibly organized tissue.
30 feet of it, yeah.
The stat pearls, sources, break down the actual microscopic architecture of the gut wall into 4 distinct layers. If you are sitting there listening to this, this is what the wall of your own stomach and intestines looks like right now, from the inside out.
So the innermost layer, the part that actually makes physical contact with the food you eat, is the mucosa.
And it isn't smooth, is it? I mean, I always
Yeah.
picture the inside of the intestine like a rubber hose, but the textbook makes it sound more like a microscopic shag carpet.
A shag carpet is a really great way to visualize it. The mucosa is highly folded. It's covered in these tiny finger-like projections called villi,
Right.
and those villi are covered in even smaller projections called microvilli.
Why so much folding, though? Why not just have it be smooth?
Surface area. If your intestine was a smooth rubber hose, food would slide right through without touching the sides enough to be absorbed.
Oh, I see.
By folding the tissue millions of times over, you massively increase the area available to pull nutrients into your bloodstream. The mucosa also houses the exocrine glands that secrete mucus to keep things sliding, and the endocrine glands that release hormones.
Okay, so that's the inner lining. Moving outward, wrapping around the mucosa, we have the submucosa.
The second layer.
Right. This is a thick layer of connective tissue. This is where all the plumbing lives. You have massive networks of blood vessels and lymphatic vessels.
Including those lacteals.
Yeah. These highly specialized lymphatic capillaries called lacteals, whose only job is to transport the heavy fats you absorb. But this is also where we find the first part of that embedded nervous system,
Yes, a Meissner plexus.
Meissner plexus.
It's a major network of nerve fibers living right there in the submucosa, constantly monitoring the chemical makeup of the food and controlling the local blood flow and gland secretions.
Moving one layer further out, we hit the heavy machinery you mentioned earlier, the muscularis externatus.
This is where the physical work of digestion happens. In most of your gut, this consists of 2 major layers of smooth muscle working in tandem.
Okay.
You have an inner circular layer that wraps around the diameter of the tube, kind of like a series of rings.
Right.
And then wrapping around that, you have an outer longitudinal layer that runs lengthwise along the tube.
And sandwiched directly between those 2 muscle layers is the second major nerve network, the myenteric, or Auerbach plexus.
Exactly.
So you have the Meissner plexus deeper in monitoring the chemicals, and the Auerbach plexus out here controlling the actual physical flexing of the muscles.
You've got it. And finally, wrapping the entire outside of the organ is the serosa, or the adventitia.
Which is like a protective layer.
Right. It is a slippery, lubricating outer membrane that ensures your stomach and intestines can aggressively churn and slide against each other in your abdominal cavity without creating massive friction and tearing.
Though looking at these rules of architecture, the stomach itself is kind of a rebel, isn't it? The sources point out a really cool anatomical quirk here.
Oh, it definitely is.
Because the rest of the tract uses those 2 muscle layers, but the stomach has a secret weapon.
Well, the stomach is an incredibly hostile environment by design.
Yeah.
It requires a massive amount of mechanical force to do its job. So, while the intestines only have the circular and longitudinal muscle layers, the stomach evolves a third layer,
The inner oblique muscular layer,
specifically designed for the stomach's workload.
which runs diagonally across the other 2.
Exactly. Because the stomach's job isn't just to push food forward, its job is to act like a violent biological cement mixer.
A cement mixer, wow.
It has to twist, wring, and churn a solid piece of food, mixing it with concentrated acid until it turns into a soupy, semi-liquid slurry called chyme. You need that third oblique angle to get the necessary twisting motion.
So if I'm trying to visualize this whole structure, I'm thinking about a modern, high-end smart home.
Okay, I like that.
The inner mucosa, the shag carpet,
Yeah.
is the drywall and the interior rooms where you actually live.
Mm.
The submucosa is the space between the walls where all the physical plumbing and electrical wires run. The muscularis externa is the structural steel beams and the motorized motors that, open the garage door.
Perfect.
But what strikes me is that this house doesn't just have wiring. It has its own localized, built-in computer servers. The Meissner and Auerbach nerve plexuses.
Mm-hmm.
They are embedded right into the walls,
Mhm.
constantly monitoring the environment and sending signals.
That localized computing power is the key to the whole system.
But wait, this is what I don't understand about human physiology sometimes. Why build a whole separate server farm down in the tissue?
Why not just use the brain?
Right. We have a brain. We have a massive central nervous system up in the skull. Why can't the brain just micromanage the gut directly? Why build a second brain at all?
To understand why, you really have to think about the sheer data load required to digest a single meal.
The data load.
Yeah.
Let's say you need a slice of pizza. As that pizza moves through roughly 22 feet of your small intestine, the tissue has to monitor everything in real time.
Everything. It has to measure the exact pH. It has to detect whether there is more fat or more carbohydrate present. It has to measure how far the walls of the intestine are physically stretching. It has to release specific enzymes at exact moments. And it's doing this continuously, inch by inch, for hours.
So if the brain had to process all of that.
The system would crash.
Really?
Oh, absolutely. If your central nervous system had to consciously or even subconsciously receive every single chemical shift and stretch receptor signal from 30 feet of tissue, calculate the correct enzyme response, and send a signal back down to fire a specific muscle fiber, it would be wildly inefficient. The latency would be too high.
It would be like the CEO of a multinational corporation demanding to personally read and approve every single email sent by every entry-level employee before it goes out.
The company would paralyze itself. Right. Exactly. So, biology delegates, it is vastly more efficient to put the processing power directly at the site of the action. The enteric nervous system handles all the day-to-day operations, the moment-to-moment adjustments, the localized enzyme releases, the muscle twitches.
So what does the brain do then?
The brain, up in your skull, only gets involved for broad, sweeping directives. It sends a vagus nerve signal saying, we are in extreme danger, shut down all digestion immediately, or we are resting, ramp up the motility.
Fight or flight stuff.
Exactly. But the actual execution of those orders is handled completely in-house by the gut.
So we have built the biological smart home. We have the walls, the plumbing, and the local servers.
Right.
But a house still needs electricity flowing through the wires to actually make the motors run. Food doesn't just fall through your 30 feet of intestines by gravity.
No, it definitely doesn't.
It has to be actively, forcefully pushed against friction. And the mechanism behind this is wild. It brings us to these fascinating structures called the pacemakers of the gut.
The interstitial cells of Cajal, or ICCs?
Interstitial cells of Cajal.
Yes, they are named after Santiago Ramón y Cajal. He was a brilliant Spanish pathologist who actually won the Nobel Prize. He was peering through early microscopes and identified these unique star-shaped cells interwoven with the smooth muscle.
And what blew my mind in the sources is where they come from.
The embryology again?
Yeah. We just talked about how the nerves come from the ectoderm, that migrating neural crest, but the ICCs actually derive from the mesoderm. They are technically a specialized type of smooth muscle cell, not a true nerve cell.
But they act like the bridge between the two.
Exactly.
They are the crucial interface. The ICCs embed themselves into the smooth muscle layers, and they connect to the surrounding muscle cells via structures called gap junctions.
Gap junctions. Those are basically microscopic tunnels connecting two adjacent cells, right?
That's a great way to visualize it. They are actual physical pores that link the cytoplasm of one cell directly to the next. This allows electrical ions to flow freely and instantly from the ICC directly into the surrounding muscle without having to wait for a chemical neurotransmitter to cross a gap.
And what exactly is the ICC sending through those tunnels?
Electricity. Specifically, a spontaneous rhythmic electrical pulse. Just like the pacemaker cells in your heart generate a continuous rhythm to keep your heart beating, the ICCs generate a steady rhythm of electrical waves in your gut.
And that has a specific name, right?
Yes, this is known as the basal electrical rhythm, or BER.
Basal electrical rhythm. And the pacing of this rhythm changes depending on where you are in the house.
It does.
The stomach ICCs are the slow, steady bass drum. They pulse about 3 times per minute. But the moment you cross into the duodenum, the very first part of the small intestine, the tempo shoots up. The ICCs there are firing up to 12 times per minute.
A huge jump.
Huge.
Yeah.
And then as you move further down into the ileum, the end of the small intestine, it slows back down to about 9 times per minute.
And the large intestine is even slower, usually only generating a pulse about once every 30 minutes. Though it ramps up significantly 3 to 4 times a day for what gastroenterologists politely call mass movements.
Which is the body preparing to literally go to the bathroom.
Right.
But here is the mechanism I want to make absolutely sure we get right. Because the Wikipedia and stat pearl sources spend a lot of time on this exact distinction. These electrical pulses from the ICCs, they're called slow waves.
Yes.
But a slow wave is not a muscle contraction.
Not at all. And that is a vital distinction. The smooth muscle in the gut operates as a syncytium.
A syncytium.
Right, which is just a fancy way of saying all the cells are electrically coupled together through those gap junctions we mentioned. When the ICC fires, it creates a slow wave. But a slow wave is just a partial depolarization of the muscle cell's membrane.
Let's break down depolarization for a second. Cells normally sit at a negative electrical charge, right? The resting state.
Yes, normally around negative 65 millivolts, which is maintained by pumping sodium ions out and potassium ions in. When a slow wave washes over the cell, it temporarily opens some ion channels, allowing positive ions to drift in, and the cell's internal charge rises slightly, maybe up to negative 45 millivolts.
So that gets a little less negative.
Right. But then it drifts back down. The membrane potential is just gently oscillating up and down.
So the electricity is humming, the charge is shifting, but the muscle isn't physically flexing at all.
Correct. The slow wave itself does not pack enough of an electrical punch to reach the critical threshold required to make the muscle actually contract. For a physical contraction to actually happen, the cell has to be hit with a true action potential.
Which in the gut is called a spike potential.
Exactly.
Okay, let's use an analogy to map this out because the millivolts can get abstract pretty quickly.
Go for it.
Imagine you were at a massive football stadium.
Yeah?
The interstitial cells of Cajal are the hyperactive fans in the front row trying to start a wave.
Okay.
The basal electrical rhythm, the slow wave, is the Mexican wave traveling continuously around and around the stadium. The wave is always moving, but here is the catch. The crowd is dead silent. They are standing up as the wave passes and sitting back down, but nobody is making a single sound. That is the slow wave. The electrical charge goes up and it goes down, but no actual work is being done.
I see where you're going with this. So what triggers the actual noise?
A chemical signal. A hormone or a nerve fires a signal into a specific section of the crowd. Now, when the silent Mexican wave reaches that specific section of the stadium, the fans don't just stand up, they scream at the top of their lungs.
And that scream is the spike potential.
Yes. Biologically, that chemical signal raises the baseline voltage of the cell just enough so that when the slow wave rides through, it pushes the cell past the threshold.
And when it crosses that threshold, the biological scream is the sudden opening of L-type voltage-dependent calcium channels.
Calcium channels?
Right. Calcium violently floods from the outside of the cell into the interior. And calcium is the ultimate trigger for the actin and myosin proteins inside the muscle to physically slide past one another.
So the muscle contracts.
The muscle contracts.
And the beauty of this system is that the scream, the contraction, can only happen if the Mexican wave is currently passing through your section.
Exactly. The ICCs and their slow waves dictate the timing. They dictate when a contraction is allowed to happen. You can only contract 3 times a minute in the stomach because a wave only passes 3 times a minute.
Right.
But the localized nervous system and the hormones dictate whether the contraction will actually happen on this pass and how forceful it will be.
So when these contractions do finally trigger, what do they actually look like?
The sources outline 2 completely different types of movement: peristalsis and segmentation. Peristalsis is the famous one.
Peristalsis is pure propulsion. Imagine a bolus of food moving into the intestine. As it physically stretches the wall of the gut, stretch receptors fire, triggering that spike potential we just talked about.
Yes.
The circular muscles immediately behind the food contract, pinching the tube shut so the food can't go backward. At the exact same time, the longitudinal muscles contract, shortening the tube. And crucially, the muscles just ahead of the food relax, opening the pathway. It is literally the biological equivalent of squeezing a tube of toothpaste from the bottom up.
That's it, exactly.
A coordinated, forward-moving ring of pressure. But what surprised me is that peristalsis isn't just a stomach thing. You find this exact same mechanism all over the human body.
Oh yeah, it is a universal biological tump. Your ureters use peristalsis to milk urine from your kidneys down into your bladder against the pressure of the surrounding organs. Your bile ducts use it.
Wow.
It even occurs in the uterus. In fact, during ovulation, reverse peristalsis in the uterus actively helps direct rapid sperm transport toward the dominant follicle.
That is wild. Reverse peristalsis.
Yeah.
Okay, so peristalsis moves things forward. What does segmentation do?
If peristalsis is a conveyor belt, segmentation is a washing machine.
A washing machine.
Yes. Instead of a coordinated wave pushing forward, segmentation involves localized, alternating rings of contraction, pinching the gut back and forth. It squishes the chyme, breaks it apart, sloshes it together, and squishes it again.
Why not just push it straight through?
Because digestion is a chemical process. You have to physically mix the food with the digestive enzymes, and more importantly, you have to ensure that every single particle of food eventually gets pushed up against the mucosal wall so the nutrients can be absorbed.
Oh, right.
If you just pushed it straight through in a clump, the food in the center of the clump would never touch the walls.
Makes total sense. And we also have to mention the housekeeper waves, because this is happening to you, the listener, right now if you haven't eaten in a few hours. The migrating motor complex.
The MMC is fascinating. When you are fasting and your stomach and small intestine are completely empty of food, the system doesn't just power down. Roughly every 90 minutes, a massive, intense wave of peristaltic contraction starts in the stomach and aggressively sweeps all the way down through the entire small intestine.
It's literally like a physiological broom.
Precisely. Its job is to sweep up all the leftover undigested debris, the sleet off dead mucosal cells, and most importantly, the resident bacteria, and aggressively flush them all through the one-way valve into the large intestine.
And if that doesn't happen, let's play out a what-if scenario. What if someone is, say, on a constant IV nutrient drip in a hospital and never actually fasts to trigger the MMC?
Well, if you didn't have these housekeeper waves, or if they were suppressed, the bacteria in your gut would rapidly multiply and migrate upward. You'd get something called SIBO, small intestinal bacterial overgrowth.
Yikes.
Yes, leading to severe bloating and malabsorption because the bacteria start eating your nutrients before you can.
Which brings us to the dark side of all of this. What happens when this exquisite electrical grid breaks down entirely? What happens when the wiring is faulty?
The pathologies are severe.
Yeah, the StatPearl's clinical notes go deep into a few specific pathologies, and they are terrifying. Let's look at Hirschsprung disease.
We mentioned earlier that the enteric nervous system is formed by neural crest cells crawling down into the gut during the 3rd week of embryonic development.
Right, the migrating cells.
Hirschsprung disease is a congenital condition where that migration process simply stops prematurely.
For some genetic reason. The sources note it is often associated with trisomy 21.
Yes, the migrating cells just run out of steam. They never make it to the very end of the colon, the rectum.
So you have a segment of the large intestine, maybe a few inches long, that is completely devoid of the myenteric and submucosal nerve plexuses. There is no local computer server there.
And without the nerves, what do you think happens?
It just doesn't move.
Actually, remember that the default state of the smooth muscle is to be slightly contracted.
Oh.
The nerves constantly send inhibitory signals to allow the muscle to relax and open up for the peristaltic wave. Without those nerves, the distal colon is locked in a state of permanent spasm.
It can't relax.
It cannot relax. So a powerful peristaltic wave pushes stool down the colon. It hits this rigid, contracted dead zone, and it just stops.
Oh, man.
The stool backs up. The colon above the dead zone stretches to massive proportions, creating what we call a toxic megacolon. It is life-threatening and almost always requires surgical removal of the aganglionic segment.
That is a mechanical roadblock at the bottom of the system. But you can also have an electrical failure at the very top. Achalasia.
Achalasia occurs right at the border where the esophagus meets the stomach. There is a ring of muscle there called the lower esophageal sphincter.
The LES.
Right. When you swallow, that sphincter is supposed to relax, let the food drop into the stomach, and then instantly snap shut to prevent stomach acid from splashing back up. But in achalasia, the inhibitory neurons in the Auerbach plexus that tell the sphincter to relax are damaged or destroyed, often by an autoimmune response.
So you swallow, the peristaltic wave pushes the food down your throat, but the door at the bottom just refuses to open.
Exactly.
The food literally just stacks up inside your chest, stretching your esophagus out like a balloon. Patients can't even keep liquids down.
And finally, a pathology that directly targets the pacemakers themselves. Gastroparesis.
This one is increasingly common, isn't it?
Unfortunately, yes, because it is heavily linked to diabetes. Let's do another what if. What if you have a patient, maybe elderly, who has had poorly controlled diabetes for 20 years?
Their blood sugar has been high.
Right. If a patient's blood glucose levels remain chronically elevated over years, the sugar becomes toxic to the nervous system and the cellular structures of the body. In diabetic gastroparesis, we see a physical documented die-off of the interstitial cells of Cajal.
The pacemakers just die. The stadium loses the fans trying to start the Mexican way.
Exactly. The stomach loses its basal electrical rhythm. The 3-beat per minute slow wave just vanishes.
Wow.
And without the slow wave, the chemical signals have nothing to piggyback on. The stomach basically becomes paralyzed. Food just sits there for hours or days, fermenting, leading to profound nausea, severe bloating, and uncontrollable vomiting.
It is a devastating reminder that mechanical digestion is entirely dependent on these electrical rhythms. But as we established with the stadium analogy, the electrical rhythm is just the baseline.
Right, it's just the timing.
The ICCs keep to time, but they still need someone to hand them the sheet music. They need the software commands that tell the system whether to speed up, slow down, dump acid, or release enzymes. And those commands are written in chemicals.
We're moving from the electrical grid to the chemical factory of the hormones.
Chemical symphony. The coordination required here is honestly staggering, because digestion doesn't just happen all at once. The body breaks it down into 3 highly coordinated phases. And the crazy part is, the very first phase doesn't even happen in your stomach.
It happens in your head.
Right, the cephalic phase.
Yes.
If I ask you, the listener, right now, to close your eyes and imagine a massive, hot, incredibly salty, perfectly fried potato chip.
Oh, I'm already there.
You can hear the crunch, you can smell the grease. Just by me talking about it, your body is changing. Your central nervous system is firing down the vagus nerve. Your mouth starts generating saliva, and your stomach literally starts pumping out hydrochloric acid, even though you haven't eaten a single thing.
It is a purely neural response. The brain is anticipating the arrival of food and prepping the factory floor. But once you actually eat that chip and it hits the stomach, we transition into the gastric phase.
Now the system goes into overdrive. The physical stretching of the stomach walls triggers stretch receptors. The sudden drop in acidity, because the food is diluting the baseline stomach acid, triggers chemical sensors. It initiates a massive local cascade of neural reflexes and hormonal release.
And this continues right into the intestinal phase. As the stomach furiously churns that chip into soupy chyme, it slowly squirts it through the pyloric valve into the duodenum.
Okay.
Now, the duodenum has a massive coordination problem. It has to suddenly pause the stomach so it doesn't get overwhelmed. It has to signal the gallbladder to release bile to handle the fat from the chip. And it has to wake up the pancreas to release digestive enzymes.
And it orchestrates this entire massive ballet using hormones produced by specialized cells called enteroendocrine cells.
These are arguably some of the most fascinating cells in the human body. They are essentially microscopic chemical factories scattered seemingly at random throughout the mucosal lining of the stomach and intestines.
But they aren't random at all. The deep dive embryology source explains exactly how these factories are built, and it is a masterpiece of genetics.
It really is.
Deep down in the crevices of the intestinal lining, in areas called the crypts, you have pluripotent stem cells. These are blank slate cells. As they naturally migrate up the wall of the crypt to replace old tissue, they encounter a gauntlet of specific genetic switches known as transcription factors.
And just to clarify, a transcription factor is a protein that binds to DNA and turns specific genes on or off. It dictates what that blank slate cell will ultimately become.
Right. And the sources trace the exact sequence. First, the stem cell gets hit with a transcription factor called MAth1.
MAS1.
MAth1 flips a switch that says, you are no longer going to be a standard absorptive cell. You are going into the secretory business.
It narrows the cell's destiny.
Then, as it keeps moving, it gets hit by neurogenin 3. This switch says, you aren't just going to secrete mucus. You are specifically going to secrete endocrine hormones.
Okay.
And finally, a factor called NeuroD1 kicks in, which literally arrests the cell cycle. It locks the cell into its final form. It commits to becoming a permanent enteroendocrine factory.
And depending on exactly where that cell ends up living, whether it's the stomach, the upper intestine, or the lower intestine, it will differentiate into a specific subtype of factory producing a very specific hormone.
Right.
We classify them by letters. You have G cells, I cells, S cells, K cells.
It sounds like an alphabet soup, honestly. But the vast majority of your entire digestive regulation is handled by just two structural families of these hormones. The first major family is the gastrin-CCK family.
Let's look at gastrin first. Gastrin is primarily produced by G cells located deep in the pits of the stomach lining. Its primary trigger is the physical presence of peptides and amino acids, basically protein. When you eat a steak, the G cells sense the protein and release gastrin directly into the bloodstream. The blood carries it back around to the stomach where it binds to specialized parietal cells.
And what does it tell those parietal cells to do?
It commands them to pump out massive volumes of highly concentrated hydrochloric acid to dissolve the steak. Interestingly, gastrin also has a powerful trophic effect.
What does that mean?
It acts as a growth factor. It stimulates the mucosal cells of the stomach to multiply and maintain a thick, healthy lining to protect against the very acid it just summoned.
That's incredibly smart. Now, gastrin's sister hormone in this family is CCK, which stands for cholecystokinin. CCK is produced by eye cells in the duodenum and the jejunum. When the fatty acids and proteins from that steak finally make it out of the stomach and into the intestine, the eye cells freak out and release CCK.
And CCK is a multitasker. The name cholecystokinin literally translates from Greek to move the bile sac.
The bile sacs, the gallbladder.
Right. CCK travels through the blood to the gallbladder and forces it to aggressively contract, squeezing a bolus of dark green bile down into the intestine to emulsify the heavy fats you just ate.
Oh.
Simultaneously, it acts on a valve called the sphincter of Oddi, forcing it to relax so the bile can actually enter the tube.
But it doesn't stop there. It also hits the pancreas, triggering a massive flood of digestive enzymes like lipases to break down the fat and amylases to break down carbs. And, crucially, it sends a signal back up to the stomach saying, hey, we have a lot of heavy fat to deal with down here. Close the valve and slow down your emptying so we have time to process this.
It is the ultimate traffic cop of the upper intestine.
But I found a detail on the StatPearls molecular breakdown that really threw me. Gastrin and CCK belong to the same family because they are molecularly almost identical.
They are very similar, yes.
At their active binding site, they share the exact same sequence of 5 C-terminal amino acids. In biochemistry, the shared sequence is called pentagastrin.
They essentially use the exact same key to try and open cellular locks.
Right. So if gastrin and CCK are wandering through the bloodstream carrying the exact same chemical key, wouldn't they constantly trigger each other's receptors by mistake? I mean, why doesn't gastrin accidentally trigger the gallbladder? Why doesn't the gut just descend into chemical cross-firing chaos?
That is the beauty of evolutionary refinement. They share the same base sequence, yes, but the receptors they bind to have evolved subtle structural requirements to tell them apart.
Okay.
We have two main receptors, the CCK1 receptor, which was formerly known as CCKA, and the CCK2 receptor, formerly CCKB.
How do they differentiate the keys?
Well, CCK primarily targets the CCK1 receptors on the gallbladder and the pancreas. Gastrin targets the CCK2 receptors on the stomach. Now, because of that shared pentagastrin tail, they can technically cross-react.
They can.
Yes, CCK can bump into a stomach receptor and cause a very weak release of acid, but the affinities are radically different. For the CCK1 receptor on the gallbladder to fully activate, it doesn't just need the 5-amino acid tail, it requires a specific modification on the larger CCK molecule called a sulfated tyrosine residue.
A sulfated tyrosine residue.
Right. Gastrin doesn't have that sulfate group in the right spot. So gastrin is functionally impotent at the gallbladder, even though it has the right tail.
It is a system of overlapping affinities, but extreme specific potencies. That is such an elegant biological failsafe.
It really is.
Okay, so that is the gastrin-CCK family handling the heavy lifting of acid and enzymes. The second major group is the secretin family, and this includes secretin, obviously, but also hormones like GIP and GLP-1.
Secretin is the great neutralizer. It is produced by S cells in the duodenum. Its primary trigger is not food, but acidity.
Okay.
When the highly acidic chyme from the stomach drops into the duodenum, the pH plummets, sometimes dropping below 4.5. The S cells detect this caustic threat and release secretin.
It acts like a fire extinguisher.
Exactly. Secretin races to the pancreas and the local Brunner's glands and forces them to dump massive amounts of water and alkaline bicarbonate into the intestine.
To put out the fire.
Right. The bicarbonate physically neutralizes the stomach acid, raising the pH back to a safe level so the delicate intestinal lining doesn't ulcerate and burn. It also sends a signal back to the stomach to stop producing gastrin.
Then you have GIP, produced by K cells further down. GIP responds directly to the presence of glucose, fats, and amino acids. GIP acts as an incretin. The moment sugar hits your intestine, GIP rushes to the pancreas and says, hey, a massive sugar spike is about to enter the bloodstream. Start pumping out insulin now so we are ready for it.
It is a brilliant anticipatory mechanism. And finally, you have GLP-1, produced by L cells even further down the tract in the ileum.
GLP-1 acts as the emergency brake.
The ileal brake, yes. If the digestive process has somehow failed and massive amounts of unabsorbed, undigested nutrients manage to make it all the way down to the end of the small intestine, the L cells release GLP-1.
What does that do?
It sends a powerful overriding signal to the entire upper gastrointestinal tract to basically freeze. It stops stomach emptying, it slows down peristalsis, essentially buying the body more time to salvage and absorb those lost nutrients.
The coordination is breathtaking. But to really appreciate how delicate this chemical balance is, you have to look at what happens when the feedback loop breaks. Let's look at the pathology of Zollinger-Ellison syndrome.
Zollinger-Ellison is a masterclass in runaway chemical cascades. In this syndrome, a patient develops a neuroendocrine tumor known as a gastronoma.
A gastronoma.
These tumors usually form in the duodenum or the pancreas. And the tumor cells are essentially rogue G cells. They uncontrollably secrete massive, unregulated amounts of gastrin into the blood.
And we know gastrin's only job is to command the stomach to make acid, so the stomach just goes into absolute overdrive.
Exactly. The parietal cells respond to this flood of gastrin by pumping out an ocean of hydrochloric acid. The patient quickly develops severe recurrent peptic ulcers all over their stomach and duodenum because the acid is literally eating through the tissue.
But the craziest part of this disease isn't the ulcers. It is the downstream effect on the rest of the digestion.
Right. That massive volume of concentrated acid, it doesn't just stay in the stomach, it pours into the small intestine. It completely overwhelms the secretin system.
The fire extinguisher can't keep up.
Exactly. The pH of the duodenum drops so incredibly low that the biological environment becomes toxic to the enzymes trying to do their job.
Because the enzymes are proteins, right?
Yeah.
And extreme acid denatures proteins. It unravels their shape.
Exactly right. The pancreatic lipases, the enzymes responsible for breaking down fat, they are dumped into the intestine, but the acid immediately denatures them. They lose their 3D shape and become totally useless.
Wow.
Because the lipase is destroyed, the patient physically cannot digest fat. The fat passes straight through their system, leading to a condition called steatorrhea, which is the excretion of massive, foul-smelling amounts of fat in the stool.
All because of one rogue tumor pumping out one hormone.
Yeah.
And doctors have a really clever, almost paradoxical way of diagnosing this, don't they?
Yes, using the secretin stimulation test. In a normal human, if you inject secretin into their blood, it inhibits gastrin release. It's the fire extinguisher.
Right.
But the cells of a gastronoma tumor are mutated. Curiously, when they're exposed to secretin, it actually stimulates them to release even more gastrin. So if a doctor suspects Zollinger-Ellison, they inject secretin. If gastrin levels plummet, the patient is healthy. If gastrin levels skyrocket, they have a tumor.
And it isn't just tumors that disrupt this loop. The sources mention that incredibly common things can throw this delicate balance into chaos. Take H. pylori, the bacteria that causes stomach ulcers. It literally destroys the acid-producing parietal cells.
Right.
So stomach acid drops. The body senses the low acid, panics, and pumps out massive amounts of gastrin to try and fix it, creating a chronic state of hypergastrinemia.
And you see the exact same thing happen when people chronically overuse proton pump inhibitors, strong antacid medications. You artificially block the stomach's ability to make acid. The chemical sensors detect the lack of acid, and the G cells just keep screaming, pumping out more and more gastrin to overcome the blockade. It highlights how relentlessly these hormones strive for homeostasis.
Now, sitting here today, we just casually list these off: gastrin, CCK, secretin. But looking at the historical archives we pulled, discovering these invisible chemical messengers completely upended the scientific world. It caused legitimate drama in the early 1900s.
Oh, it was a massive paradigm shift. You have to understand the prevailing dogma of the time. Prior to 1902, the undisputed king of physiology was Ivan Pavlov.
Yes, the man with the dogs and the bells.
Exactly. Pavlov had established that the nervous system controlled everything. The scientific consensus was nervism. They believed that all digestive organs were hardwired directly to the brain, and that physical reflexes traveling through the nerves were the sole trigger for releasing digestive juices.
If the brain didn't command it through a wire, it didn't happen. That was the rule.
That was the absolute rule. But then, in 1902, two British physiologists, William Bayliss and Ernest Starling, set up a legendary experiment that shattered Pavlov's entire worldview.
Let's set the scene. They are in a laboratory in London. They want to figure out exactly how the pancreas knows that food has arrived in the duodenum. How does the timing work?
Right.
So they take an anesthetized dog.
Right.
And they do something incredibly meticulous. They surgically isolate a loop of the dog's duodenum, and they systematically sever every single nerve connecting that intestine to the brain, the spinal cord, and the pancreas.
It is a complete neural blockade. The pancreas is now totally physically isolated from the central nervous system. Under Pavlov's rules, that pancreas should be dead to the world. It should not be able to receive any signals.
But then Bayliss and Starling introduce a dilute hydrochloric acid solution into that isolated loop of intestine, simulating the arrival of acidic chyme from the stomach.
And to the absolute astonishment of everyone in the room, the denervated pancreas immediately springs to life and starts pumping out alkaline digestive juices.
Which is impossible under nervism.
Exactly. Because the wires were cut, the signal couldn't possibly be electrical. The only remaining physical connection between the intestine and the pancreas was the bloodstream. They realized, in that moment, that the intestine must be secreting an invisible chemical messenger directly into the blood, which travels to the pancreas and unlocks a response.
They realized they had discovered an entirely new method of biological communication. They scraped the mucosa of the intestine, extracted the chemical, proved it worked by injecting it straight into a vein, and named this mysterious substance secretin.
And 3 years later, in 1905, Starling gave a famous lecture where he officially coined a new word to describe this entire class of chemical messengers. He took the Greek root hormone, which means to excite or to set in motion, and gave us the word hormone.
But, and I love this detail from the historical sources, the textbooks usually get the timeline slightly wrong.
They do.
Because of Starling's famous lecture, nearly every biology textbook calls secretin the first hormone ever discovered. But that is a massive historical misconception.
It is a point of deep historical pedantry, but you are right. Back in 1894, 8 years before Bayliss and Starling, two other British researchers, George Oliver and Edward Albert Schaefer, were grinding up adrenal glands from animals. They injected the adrenal extract into dogs and observed a massive instantaneous spike in blood pressure.
They had discovered adrenaline, epinephrine.
Precisely. They published their full findings in 1895. So adrenaline was definitively the first hormone discovered and isolated. Secretin was just the first one to be called a hormone because the word didn't exist when adrenaline was found.
The history of science is so messy. And it gets even messier when we look at how they discovered CCK. It is an absolute comedy of errors.
Oh, the Pancreozeman saga?
Yes. In 1928, researchers Ivy and Oldberg isolate a hormone that causes the gallbladder to contract. They name it cholecystokinin, CCK. Simple enough. But then, 15 years go by. It's 1943. Two different researchers, Harper and Raper, isolate what they firmly believe is a completely different hormone that stimulates the pancreas to release enzymes. They confidently name their new discovery pancreozemin.
And for decades, the entire scientific and medical community treated CCK and pancreozemin as two entirely distinct, separate hormones. Textbooks had completely separate chapters for them.
Until the 1960s.
Right. In 1968, two Swedish biochemists, Johannes Erik Jorpis and Victor Mutt, decided they want to definitively map the chemical structure of these hormones. They painstakingly isolate and purify massive amounts of porcine, meaning pig, cholecystokinin.
Right.
It takes years of rigorous chemical processing to strip away all the impurities until they have the pure peptide sequence.
And when they finally analyze the amino acid structure of CCK and compare it to the structure of pancreozemin, they realize it is the exact same molecule.
The exact same string of amino acids.
The scientific community had been studying the exact same hormone for 40 years under two different names, completely unaware that it just had two different jobs.
I think it beautifully illustrates the reality of the scientific method. Knowledge is iterative, It is rarely a sudden perfect stroke of genius.
Yeah.
You have multiple brilliant researchers working in different decades with different tools, essentially stumbling around in the dark.
Like the blind men and the elephant.
Exactly. They're like the blind men touching different parts of the same elephant. Ivy and Oldberg touched the trunk and called it CCK. Harper and Raper touched the tail and called it pancreosemen. It took 40 years of technological advancement in peptide sequencing to turn on the lights and realize it was just one elephant.
I love that. Okay, so we've mapped the physical structure of the smart home, we've traced the electrical grid, and we've explored the dramatic history of the chemical hormones. But now we have to look at the most mind-bending part of this entire deep dive. We have to cross the blood-brain barrier.
This is where it gets really strange.
Because these hormones we just spend so much time on, they don't just stay in your digestive tract. They cross over into your neurology. They bridge the gap between what you are digesting and how you are actively feeling. Let's talk about the gut-brain axis.
This is where the concept of the second brain drops the metaphor and becomes completely literal. The enteric nervous system, with its 100 million localized neurons, is not an isolated island. It is in constant bidirectional communication with the central nervous system.
The primary superhighway for this data is the vagus nerve, right? A massive cable running straight from your abdomen up into your brainstem.
Yes, and the traffic on that highway is heavy. But what is truly paradigm shifting is looking at the chemical neurotransmitters involved. We have spent our whole lives associating chemicals like serotonin and dopamine entirely with the brain. Serotonin is the happiness and well-being chemical. Dopamine is the reward and motivation chemical. When someone is depressed, we treat their brain.
But the Kaiser Permanente article highlights a statistic that is just staggering. According to the research, 90% of the body's entire supply of serotonin and 50% of its dopamine are not produced in the brain at all. They are produced down in the gut.
The specialized enterochromatin cells in the gut lining, interacting constantly with your gut microbiome, are churning out massive quantities of these mood-altering neurotransmitters. They use them locally to stimulate motility, but the systemic implications for mood regulation are immense.
And it isn't just serotonin. The digestive hormones we just talked about have secret double lives in the brain. Let's look at the dark side of CCK.
Ah, yes.
Down in the gut, CCK is the helpful traffic cop digesting your fats. But up in the central nervous system, CCK is the most abundant neuropeptide present. It is heavily concentrated in the limbic system, the hippocampus, the amygdala, the areas of the brain that control fear, emotion, and memory.
And its function in those fear centers is profoundly different than in the gut. In the brain, CCK is highly anxiogenic. It generates anxiety. In higher doses, it is purely panicogenic.
It causes pure panic. The sources note that this effect is so reliable, so mathematically certain, that neurological researchers literally use a synthetic fragment of it, the CCK4 tetrapeptide, to intravenously induce massive panic attacks in completely healthy human test subjects.
It sounds barbaric, but it's true.
They just inject them with CCK4 so they can test how well new anti-anxiety drugs work.
It's incredible. When they put these test subjects into PE scanners and inject the CCK4, they see massive immediate shifts in blood flow directly to the anterior cingulate gyrus and the amygdala. The brain's fear circuitry just lights up like a Christmas tree.
Wow.
Furthermore, we are finding that malfunctions in how the brain processes CCK are directly implicated in the terrifying visual hallucinations experienced by patients with advanced Parkinson's disease.
Okay, but if I'm listening to this, I'm thinking of an incredibly obvious paradox here.
What's that?
We established that every time I eat a fatty meal, if I go to a drive-thru right now and eat a double bacon cheeseburger, my gut eye cells are going to release a massive systemic flood of CCK into my bloodstream to digest the fat.
That's right.
If CCK triggers severe panic attacks and hallucinations, why doesn't eating bacon induce a state of screaming terror? Why aren't we all panicking after Thanksgiving dinner?
You've hit on the crucial anatomical firewall that keeps us sane. The blood-brain barrier.
The blood-brain barrier.
The CCK that is manufactured in your gut, the CCK that floods your systemic blood circulation to squeeze your gallbladder, it is physically incapable of crossing the blood-brain barrier. The tight junctions of the capillaries in the brain simply will not let the molecule pass through from the blood into the brain tissue.
Oh, so the brain is completely walled off from the digestive supply.
Entirely. The brain has to manufacture its own separate, highly localized supply of CCK for its neurological functions. It synthesizes it locally as a larger precursor molecule and cuts it down into a specific active form, mostly the sulfated octopeptide CCK8S, which acts directly on the brain's synapses. The digestive pool of CCK and the neurological pool of CCK are completely segregated systems.
Thank goodness for the blood-brain barrier. But CCK isn't the only hormone doing double duty. What about the fire extinguisher? Secretin.
Secretin also has a profound role in the brain, specifically in osmoregulation, which is the critical balance of water and salt in your body. We find secretin receptors highly concentrated in the hypothalamus, particularly the paraventricular and supraoptic nuclei.
What is it doing up there?
When you become dehydrated, the concentration of salt in your blood rises. The hypothalamus detects this hyperosmolality. Secretin acts within these brain regions to trigger the release of a different hormone called vasopressin from the pituitary gland.
And vasopressin does what?
It travels to your kidneys and commands them to stop making urine, reabsorbing water back into the blood to literally save your life.
And the history of secretin crossing into the brain has its own weird, dramatic chapter. In the late 1990s, there was this massive, almost hysterical medical craze. Some anecdotal reports surfaced suggesting that intravenous injections of synthetic secretin were dramatically improving the social behavior and language skills of children with autism.
It was a theory born from the realization that gut issues are frequently comorbid with autism spectrum disorder. The idea was that fixing the gut-brain chemical axis with secretin could somehow rewire the neurological symptoms.
It got so huge, featured on national television. The parents were desperately seeking this unapproved treatment. It forced the National Institutes of Health to step in and fund massive, rigorous, double-blind, placebo-controlled clinical trials.
And those trials, which were ultimately published in top-tier journals like the New England Journal of Medicine, definitively proved that secretin was completely ineffective at treating autism. There was no clinical difference whatsoever between the secretin and the placebo.
That must have been heartbreaking for the families.
It was a harsh reminder of the power of the placebo effect. But it perfectly highlights just how desperate we are to crack the code of the gut-brain connection. We intuitively know that our gut chemistry dictates our mental state.
Which brings us to the most practical part of this whole deep dive. If this is true, if the gut has this profound, literal impact on our neurotransmitters, our physical motility, and our baseline anxiety, how do you, the listener, consciously maintain it? How do you take care of a second brain?
It fundamentally comes down to two pillars: dietary mechanics and microbiome maintenance. Because every single thing you put into your mouth directly alters both the physical movement and the chemical environment of your gut.
Let's look at the mechanics first. The Stab Pearl's motility source makes it clear that meal composition literally changes your peristalsis. We talked about how high-fat foods trigger CCK and GIP.
Right.
Those hormones act as a heavy brake on the stomach. If you eat a heavy, fatty meal, your gut intentionally slows your motility to a crawl to give the liphases time to work.
And the physical state of the food matters. Solid, complex meals require vastly slower, more forceful, muscular contractions from the stomach to physically grind them down, compared to just drinking a liquid smoothie, which empties almost immediately. You are dictating the electrical workload of your ICC pacemakers with your fork.
But the deeper, more profound maintenance is about the microbiome, the trillions of bacteria, viruses, and fungi living in your lower intestine. You are carrying around an alien ecosystem, and that ecosystem is what regulates that 90% of your body's serotonin.
You really are an ecosystem.
The Kaiser Permanente article details 4 crucial dietary components for supporting this ecosystem. First on the list, probiotics.
Probiotics are the actual live beneficial bacteria found in fermented foods. We are talking about kimchi, kefir, yogurt with live cultures, kombucha, sauerkraut.
Right, the live stuff.
Yes, when you consume these, you are directly reinforcing the population of good bacteria in your gut, which crowd out the pathogenic bacteria and physically lower systemic inflammation.
But dropping new bacteria into the environment isn't enough. They are living creatures. They need to eat. And that brings us to the second component, prebiotics.
Prebiotics are essentially the food for the probiotics. They are specific types of dietary fiber found in complex carbohydrates. Apples, bananas, oats, legumes, garlic, onions, asparagus.
Okay.
You, the human, lack the enzymes to digest this specific fiber. It passes through your stomach untouched. You aren't eating it for yourself. You're eating it specifically to deliver it to the bacteria in your colon. They ferment that fiber and thrive.
And when they ferment it, they produce short-chain fatty acids that actively heal the lining of your gut.
Exactly.
Third on the list, we have polyphenols.
Polyphenols are powerful plant-based antioxidants. You find them heavily in green tea, black tea, dark cocoa, colorful berries, and spices like turmeric.
And what do they do for the gut?
Their primary role is to reduce oxidative stress and cellular damage in the delicate mucosal lining. But what's truly fascinating is that when your gut bacteria break down these polyphenols, the chemical byproducts actually cross into the bloodstream and assist in the release of those mood-altering neurotransmitters.
And finally, omega-3 fatty acids. These are the anti-inflammatory heavyweights, found in fatty fish like salmon and sardines or in chia seeds and walnuts. They provide direct, clean energy to the cells lining the gut, suppress inflammatory chemical cascades, and help balance the diversity of the bacterial populations.
It really requires you to look at every meal as a multifaceted intervention. You are managing a complex ecosystem, not just fueling a machine.
I really like that framing. I've always thought of eating as, putting gas in a car engine. The tank is empty, you put fuel in, the engine burns it, end of story. But that's entirely wrong. Eating is much more like tending a highly sensitive exotic botanical garden. You aren't just feeding yourself, you're actively cultivating trillions of microscopic organisms that literally control the production of your serotonin levels. If you feed the garden trash, the soil turns toxic, the wrong weeds grow, the chemical balance collapses, and your mood and physical health plummet.
That is exactly the shift in perspective the science demands. You are the custodian of a biological network that rivals the brain in complexity.
We have covered so much ground today. We started with a tiny microscopic tube of endoderm folding in a 3-week-old embryo. We built the 4 structural layers of the smart home and wired it up with the electrical pacemakers of the interstitial cells of Cajal.
We did.
We explored the messy, dramatic chemical history of Bayless and Starling severing nerves to discover secret and prove that hormones exist. And we crossed a blood-brain barrier to see how the gut manufactures the very chemicals that dictate our happiness and our panic.
It is a breathtakingly intricate system, and the reality is we are still only scratching the surface of the gut-brain dialogue.
Which leaves us with a final, mind-bending thought to mull over as we wrap up. For centuries, humanity has evolved its philosophy, its religion, and its science to view the brain inside our skull as the absolute, undisputed center of our consciousness.
Sure, the seat of the soul.
Exactly. The brain is the seat of the soul, the origin of our decisions, and the source of our emotions. But consider the biological reality we just mapped out today. Our gut generates its own spontaneous, independent electrical rhythms. It possesses 100 million neurons that operate autonomously. It manufactures the vast majority of our serotonin and dopamine. It constantly beams massive amounts of sensory data up to the brain via the vagus nerve.
It's a lot of data.
So, is it possible that our core primal emotions, our baseline sense of safety, our sudden spikes of dread, our profound feelings of joy, actually physically originate in the belly, and that our massive, highly evolved cerebral cortex is merely the organ whose job is to translate those visceral chemical signals into conscious thoughts and spoken words?
It fundamentally redefines the concept of human intuition.
It really does. The next time you are standing backstage and you get that flutter, the next time you drop your keys and get that heavy pit in your stomach, the next time you make a major life decision because you decide to trust your gut, remember the science. You might not be speaking metaphorically at all. You might actually be listening to your second brain. Thank you so much for joining us on this deep dive. I know I'm going to be thinking very differently about my next meal, and I hope you do too. Until next time, take care of your garden.